And now, without further delay, I'm going to invite Norbert Ngowi. Norbert is from Mzumbe University in Tanzania. He will give a presentation about valley bottom agriculture in a changing climate and its impact on stream flow and land use in southern highlands, Tanzania. Norbert, the floor is yours.
Thank you very much. Good morning, everyone. My name is Norbert Ngowi. My presentation is on bottom valley agriculture in a changing climate and its impact on stream flow and land use in southern highlands of Tanzania. But before I proceed, I just want to give an overview of agriculture at the people ecology nexus of the southern highlands of Tanzania in 6 slides. On plate 1 is the typical farmland during the dry season, and plate 2 is the habitat of the valley bottom during the dry season. Plate 3 is the conversion of the bottom valley to fish ponds at the interface between the bottom valley and the uplands during the dry season. Plate 4 and plate 5, these are the temporary farmlands on the bottom valley during the dry and the rainy season, is being presented on plates 4 and 5. And plate 6 is the upland farms during the dry seasons. On the background, smallholder farming, mainly the rain-fed, is most common in Africa, but is instrumental for human livelihoods, accounting nearly 50% of per capita in Tanzania. Valley bottom agriculture is the system of farming practiced among smallholder farmers whose farms are smaller than 5 hectares in the southern highlands of Tanzania. This system utilizes naturally available moistures and water in small streams and short wells around the farms to produce food and income of farmers. Research on utilization of these landscapes for agriculture as climate-related risks in the ecology of the unimodal rainfall is important. However, the implication of changing the agricultural systems from valley bottom to uplands on land use and the stream flow have been lacking. This work was planned to fill this knowledge gap. The objective of this study was to understand the effect of shifting of valley bottom to upland farming systems in the selected wards within a hemi-cluster of Southern Agriculture Growth Corridor of Tanzania to specifically consider changes in the land use cover, the stream flow within a period of 30 years. The methodology. Objective 1 sought to understand the changes in the cover of major land uses. We rely on data sources from the satellite images taken between 1985 and 2015 with a spatial resolution of 30 meter by 30 meter, which were downloaded from the United States Geological Survey. And we supplemented these satellite images with the topographical maps of the study area. We used the ERIDAS Imagine 15 software in processing our data. We used the ArcGIS version 10 and ArcMap to finalize the analysis. So our interpretation of the images looked at the major land uses, which are 9, in terms of shape, size, tone of color, texture, and the pattern. Objective 2 aimed to understand changes in the stream flow, where we relied data for rainfall for the same period we took for the land uses from the Basin Water Office and the meteorological agents, but we supplemented the gaps with the climate hazard group infrared precipitation with the station data. The stream flow data for 1985 to 2015, which are gauged and monitored data, were also used. But also we supported our data with the literature sources. We used the trend version 1 of 2002, which we downloaded from Australian e-water toolkit to analyze the water by converting the annual time series to a comma delimited method. So we downloaded this to a trend screen by selecting the CSV file and test the results and give an output. Results. With respect to objective 1, which sought to understand changes in the curve of major land uses, we found that the large gains in cover of 2 major land uses, particularly the farming or cultivated land, which were found to have increased by almost 157%, and the settlement areas, which were found to have increased by 52%. With respect to the bottom valleys, we did found that there were more declines in cover along the bottom valley habitats, where open waters had declined almost by 97%, followed by flood plains. With respect to objective number 2, which sought to understand the changes in the stream flow, on the left side, figure A, we looked at the historical rainfall pattern of the study area for a period of 30 years. And we did found that of the 3 study wards, Maboga ward was found to have received lowest rainfall compared to the rest of the study wards, the Lumuli and the Ifunda wards. On chart figure B, which is on the right, the changes in the long-term rainfall flow pattern. The bar, which are in blue color, represents the rainfall pattern, while the curve in red represents the flow of the streams. We found that the nature of the peaks show that rainfall, the high rainfall did not necessarily impact the stream flow, with the exception of the period of 1997-98, when we had El Nino in East Africa, where in that year, 90 millimeters of rainfall was recorded with a flow of QMX of around 22 was recorded. However, the nature of the peaks, as it indicates on graph B, it shows that, for example, in year 2005-2006, which had almost recorded the high rainfall, but it had the lowest flow. The same is for the year 2010-2011, where we had high rainfall, but least stream flow. So we correlate the rainfall and the stream flow. On figure 1, we did a regression by excluding the outliers. And in figure 2, we included the outliers. That we found that by excluding the outliers, the results show that 37% of the variation in the stream flow is explained by rainfall, while 27% is explained by other factors. This is on figure 1 and figure 2. So the classification of these correlations shows that there is a weak correlation which was observed between rainfall and the stream flow, implying that rainfall did not significantly affect the stream flow. Discussion and conclusions. Changing climate and culture, particularly the demand for food and methods of farming were found to be the major drivers for the bottom valley to upland agriculture. On figure A, which is on the left, plate A, it shows that opening up new areas on the uplands had led to positive change in terms of increase in food production as well as income security of the small farmers. But it had a negative change in terms of forest clearance, which you can see, soil erosion indicated by the color of the water, and the siltation effect, henceforth shrinking of the agricultural land and the stream flow down the valley. These results are similar to those of Pierre and his colleagues that showed that expansion and intensification of the agriculture has become a driver in the cover of the lowland areas. As you can see on plate B, which is on the right, it shows that the farming methods of raising the camber beds has blocked water around the camber beds from returning to the mainstream, thus affecting the water flow within the major streams. These results are similar to those of Mzumbe and Dixon, who show that farming methods along valleys had reduced the stream flow. So this study had recommended a study to investigate climate smart agriculture technique in valley bottom to uplands to reduce food insecurity while protecting nature. And this, by doing this, it will find the method of returning the water which are standing around the raised camber beds, as you did see on slide number 10B, to return to the mainstream, thus not affecting the downstream flow. Thank you. This is the end of my presentation. Thank you.
Thank you, Norbert, for this presentation, and we see a lot of approval coming up from the audience. May I invite Tim Maes to come on stage, please?
Good morning.
Good morning, Tim. And Tim Maes is from KU Leuven in Belgium, and Tim is going to bring a presentation basically for laymen like me about schistosomatiasis, and he will talk about the distributional changes of the snail that is the intermediate host of schistosoma in the face of climate change. Tim, the floor is yours.
All right, let me first share my screen, and then I'll wait a few seconds for everybody to load. So, beware of the snails. Is this a sign that we will see more often in the future? Sounds plausible to some of the African colleagues, but most of the European colleagues will probably laugh at this sign. However, freshwater snails have been listed among the world's most deadly animals alongside mosquitoes. And this is mainly because of the parasites they carry and the diseases that these parasites cause. The most well-known example is malaria that is transmitted through mosquitoes. But also freshwater snails can be a host to multiple different parasite species. Furthermore, we do know that climate change is affecting these vectors, distributions, and the diseases they transmit. However, the exact mechanisms affecting these disease transmission are not really well known because we do not have a lot of ecological data on the ecology of these disease vectors. That's why I want to investigate the ecology of these freshwater snails vector and see how they respond to climate change so that we can predict if we should place these signs in Europe more often or that we can abandon them in some parts of Africa. So my name is Tim Maes and I'm a PhD researcher at KU Leuven and the Royal Museum for Central Africa in Belgium. And today I'm presenting you my work on the distributional changes of Bulinus truncatus, intermediate snail host of Schistosoma species in the face of climate change. So there are two main unknowns for most of you, I believe, in this title. And first of all is, of course, Bulinus truncatus and Schistosoma species. I will start off with Schistosoma. Schistosoma is a genus of parasitic flatworms that affects humans and animals and causes the disease schistosomiasis. Schistosomiasis is a tropical disease affecting over 240 million people worldwide and gives rise to a range of bladder and intestinal problems. Now, despite the fact that this disease affects so many people, not a lot of people in the developed world or in the global north have ever heard of this disease. And there are two main reasons for this. First of all is that it's a chronic disease. So it doesn't have a lot of acute deaths, but people do have a lot of bladder and intestinal problems throughout their lives, and high infections can even lead to infertility, stunted growth, and impaired cognitive development. Also, this gives rise to a lot of economic losses in the affected countries. The second reason why the developed world or people in the developed world have never heard of this disease is because of its distribution. As we can see on this map, the disease is mainly prevalent in Africa and some parts of South America, China, and Southeast Asia, but it remains absent in most of the global north. Now, despite that a cure is present for the disease in the form of deworming drugs, it remains prevalent all over the world, and the number of people affected has not changed that much. And the reason for this, we should have a look at the life cycle of this parasite. So in figure A here, we see the adult worms of Schistosoma, and these worms live inside of the veins of the bladder and the intestines of humans or animals. Here they reproduce sexually, and they produce eggs that are excreted outside of the human or animal body through the urine or the feces. When these eggs come into contact with fresh water, they hatch, and the first larval stage, the miracidium, here depicted in C, emerges. This larval stage then searches in the water for a suitable freshwater snail host where they can reproduce asexually. When these snails start shedding the second larval stage of the parasite, which is called the cercaria, and the second stage larvae then searches for a suitable human host when they come into contact with water. They penetrate the skin of the humans and they migrate through the blood veins towards the bladder and the intestines, and the circle is completed. Now, we do have very effective drugs for these adult worms inside of the human body. However, when we only target the humans through the mass drug administration campaigns, we oversee one important aspect of the schistosome life cycle, and that is the snails. As long as the snails stay infected with the parasites, reinfection can still occur of humans and the disease remains prevalent in an area. That's why it's also important to not only focus research efforts on humans, but also on the snails. And one of the most important snail hosts of Schistosoma is Bulinus truncatus. Bulinus truncatus is a small freshwater snail, as we can see here on the picture, that lives inside of streams, ponds, and rivers. It has a very wide distribution, as shown on this map, but ranges all across Africa and some parts of Europe and the Middle East as well. And that's exactly why it makes it so interesting subject to study the effects of climate change on Schistosoma, because it has the widest distribution of all of the schistosome intermediate snail hosts. So if you want to assess disease risk evolution in the future, then this is one of the most interesting species to investigate. Though climate change has been known to affect disease risks, and also schistosomiasis has already been shown to change its disease dynamics in the future. And we have some examples of this. So the first example is the recent emergence of schistosomiasis in Corsica. Schistosomiasis has been discovered in 2015 to be transmitted in Corsica, and it has been already proven that this disease is now persistent there. And this is mainly because the temperatures in Corsica have become warm enough for the parasites to survive there. The snails were already present before, but now also the temperatures have become warm enough for the parasites to survive. The parasites got there through increased mobility from Senegal to Corsica and has been transported through infected humans inside the new transmission area. The same goes for Spain, where new recent infection foci have occurred in areas where the snails were already present, but now also the parasite has emerged. Now, Climate change does not only have an influence on the parasites themselves, but also on the snails that transmit the parasites. And this has been shown in Africa, where in recent years we have seen a lot of droughts in Africa and not a lot of rainfall, which caused a lot of rivers to dry up almost completely, but it left a few ponds or a lot of ponds with only a few water in it. And these have become really the hotspots for snails to emerge. So these snails now thrive in these new habitats, and they also got schistosome parasites from neighboring areas. And so we also see a lot of new hotspot areas in South Africa. So there are a lot of factors influencing this disease cycle in the changing climate. And first of all, there's the effect of climate change on the parasites themselves. But we also have the increased human mobility that transports the parasites from one area to another. And then finally, we also have the effects of climate change on the snails and the distributional changes of these snails. So what I want to do in my research is to see how this distribution of these snails will change and how this will affect disease dynamics in the new areas or in the areas where it was previously present. But in order to model the distribution of these snails, we need, of course, data. And the most important data that we need is data on the tolerance limits of these snails to different abiotic factors. So the first step in my PhD was to look if this data is available and if it's readily usable in these modeling exercises. Here I will present to you the results of my study in brief. And in this plot, we can see that the number of studies that is available for each Schistosoma intermediate host species. And we see that a lot of studies have focused on Bulinus truncatus and Bulinus pfeifferi, which is good because these are the most important intermediate host species. So it's good that we have a lot of studies for them. However, if we dig a little deeper and we check at the number of studies for each abiotic factor, we see that a lot of studies have focused on desiccation and water chemistry, but only 5 have focused on temperature. And temperature has been listed as one of the most important factors influencing species distributions on a global scale. And what's more, that these 5 studies are not just focused on one single species. No, these are 5 studies for all of the species listed in the previous slide. So the distribution of the number of studies and the abiotic factor studied per study is really variable among the different species. And if we also look at the number of studies executed per country, we see that there is a lot of variability. And this also influences the usability of this data for species distribution modeling. Because a snail living in Corsica will not have the same tolerance limits as the ones in Tanzania, because Corsica is far colder than Tanzania. And we can expect that the snail species living in Corsica will have lower tolerance limits to temperature than the species in Tanzania. So we do have a lot of pieces of our puzzle already to construct these species distribution models for future climates, but we do not know exactly how they will fit in each other, how many pieces are missing, and if all of the pieces are exactly from the same puzzle. So it's really hard to do this modeling. So that is why in my future experiments that is coming up in January, I want to dig a little deeper into this ecological aspect of these snails. So at the moment, I'm having a snail culture I'm breeding snails in the lab in Leuven. And as you can see here, we do have quite a success with snail breeding. So on the left side, on the top left side, you can see the number of eggs that's laid by just a few snails and then the breeding setup. And the goal of the experiment or the experimental setup, I will discuss now briefly. So in the summer of this year, we went to different spots across France and Africa, and we collected snails there. So we collected snails from France, Senegal, and Zimbabwe. Why these 3 countries? Because these represent a strong temperature gradient, with France being the coldest, Senegal being the warmest, and Zimbabwe is somewhere in between. On top of that, we also have a latitudinal gradient, so we have snails from a lot of different localities and with the highest possible variability. These snails are now being bred in the lab in Leuven until the 2nd generation to limit effects of stressful events that happened before with the parent generation, and we don't want to get them in our experiments, so we breed them until the 2nd generation. And then we will subject them to a temperature gradient. So we have very high temperatures of 36 degrees Celsius, but we also have very low temperatures of 4 degrees Celsius. And the goal of this experiment is to construct thermal performance curves, as we can see in this figure. So a thermal performance curves plots the performance of a species on the y-axis and the temperature on the x-axis. And as we can see on this figure, the thermal performance, the performance of a species increases as temperature increases until it drops suddenly. This is quite easy to understand. If I place you without clothes on the Arctics, you will probably not survive that long. Your performance will be very low. You will probably just die. So you will have performance below zero. But as temperatures increase, you will get higher and higher on this curve, and you will perform the best around 25 or 30 degrees Celsius, probably for humans. If it gets even hotter, yeah, you will probably overheat, and you will eventually also just die, and you will have a performance less than zero. The points where we perform best is called the tolerance breadth. And this is exactly what we need for these snails as well. If we know where these snails can survive, whether they can reproduce, where their performance is above zero, we know exactly what temperatures they can resist and how they also respond to climate change. So this is what it will look like more or less in practice. So we have a lot of snails in separate cups, and we will feed them ad libitum so they don't have any restrictions on growth. And we will count their egg masses, and we have different endpoints. So the first endpoint is, of course, the size of the snails. How fast do they grow? Are there differences between the different temperature treatments, and are there differences in the different origins? Then also we have the fecundity. How much eggs do they lay, and how big are these eggs? Because this also influences the survival and the reproduction capacity of these snails. And then finally, we have the survival of the snails. How fast do they die? Are there differences between the origins and the temperatures? These are quite robust endpoints, but they only give one view on how these snails respond. For example, if a snail at 24 degrees, it's perfectly possible that it survives as good as the snails at 32 degrees. But that doesn't mean that the snails at 32 degrees perform as well as the snails at 24, because they might be just more stressed. And this doesn't have an influence on the laboratory setting, but this might influence them in real life, in the real habitats where they have competition with other species or even other snails. That's why it's also important to look at their physiology. How much fat do they contain? How much sugars? How much protein? And if we look at that, we will have a better picture on how these snails respond to different temperature and what is the influence of their origin. And finally, we will also have a look at the genetics of these snails, so their DNA. Are they genetically adapted to their local temperature, to their local climatic conditions? And if we have all these endpoints and we put them together, it is possible to construct species distribution models for the current climate. So where do they occur now? But more importantly, where will they occur in the future? So in summary, we do know that climate change affects disease dynamics, including schistosomiasis distributions, but we do not have enough ecological data on their intermediate snail hosts to really model this and predict where these snails are present. Data is very fragmented, it is incomplete, and not always so reliable. So that's why it's really needed to carry out the experiments, gather more data, and eventually predict where the snails will be present, and that we can predict if it is necessary to really place these signs more often in Europe, or that we can even abandon them in some parts of Africa as the temperatures get too warm for the snails. Thank you.
Thank you very much, Tim. So let me remember you that if you have questions, you can write them in the chat. For the moment, I have one question for Tim. I'm a geographer, so I was looking to your maps, and I saw a strong distribution of both the snail and the disease in Africa, but not very much in the neotropical areas in Latin America. And with global travel, how comes that this small snail doesn't expand easily into Latin America?
Because there are some important ways that these snails can travel. A snail is just a small animal. So it's mainly transported through river currents, some from one place to another. And we see that in Latin America, the snails are mainly situated across the ocean borders. So it's very hard for these snails to be transported upstream. However, there's one main reason that snails can travel large distances and why they occur all across Africa, for example, and some parts of Europe, and that is that they can also be transported on feet and feathers of birds. Especially the eggs are very sticky, so they stick to the feet of birds. And in Europe, we have a lot of migratory birds that transport the eggs from one place to another.
South, yes.
I don't know the situation exactly in Latin America, but I think there is a less bird migration, especially since the snails are there situated across the middle line of the earth. I cannot recall that.
Yeah.
So there's less bird migration, I believe, between the north and the south.
I have other questions coming in, and you are studying the snail.
Yeah.
But in fact, such a study, it involves both the snail and then the parasite itself. Mm-hmm. And the parasite might have other tolerance limits than the snail.
Yeah, that's indeed true.
That?
So the snails and the parasites, they do have different tolerance limits, but they do not change that much. So that's what we see in Corsica. So the snail is there almost at their northern limit, as we know now, because we haven't observed them more northward. And now the parasite has also become established in Corsica. So we do know that the tolerance limits between the snail and the parasite do not change that much. But what we do know is that if we see a more change of distribution more northward, we can expect that the parasite can also come northward, and we can prepare before the snail and even before the parasite arrives there to take the necessary precautions in those areas. But it's indeed true.
One last quick questions. Your snails are getting plenty of food in Leuven.
Yeah.
But maybe they don't have the same amount of food in their real environment. How do you handle that?
That's indeed true. It's always a question in ecological experiments. How do you feed the snails? Do you give them plenty of food? Do you restrict their food intake? In this case, we just want to see if the snails live in ideal conditions. How do they respond to climate change? What are the differences? And the reason that we do this physiology is to check on these minor differences between them. And this makes it possible to extrapolate those differences to real life, to conditions where they have competition, where they have predation, where they don't have a lot of food. But food restriction mainly affects disease or species abundance. It doesn't affect their distribution, their global distribution, but it does affect how much snails are present in a certain habitat. So that's why I believe it's important to have these snails now well fed and that they don't have any restrictions on competition, and that we can just check on the effects of temperature and just only that effect and not all the rest. That's what we do in a laboratory settings. Otherwise, we could just have go to the field and carry out the field study.
Thank you very much. Thank you very much, Tim. Now I'm going to ask David Oyo to come to the stage. David Oyo is from the University of Ibadan in Nigeria. Okay, welcome, David.
Welcome.
David is with us, and he is going to bring a presentation about climate change adaptation strategy for dry season amaranth cropping systems in the tropics. A case study from irrigated watermelon as live mulch. David, the floor is yours.
Thank you. My name is David Ojo, and I'm speaking from the farm office here at the National Horticultural Research Institute in Nigeria, Ibadan, and I will be presenting on the effect of climate change on irrigated watermelon as a text case. I'll be sharing my screen with you. I will be sharing the screen. I'm just waiting for it to come up, and then we share. Let me see if it is coming up. I've made this so simple, such that it will not consume much space. Can you hear me? The title. The title of my presentation is Climate Change Adaptation Strategy for Dry Season Amaranth Cropping Systems in the Tropics. The case study we are using here is irrigated watermelon. And my name, again, is David. And my cell phone and contact is here presented for further discussion. This investigation seeks to quantify watermelon life mulch densities, on yield potentials, and climate change adaptation capabilities. Our result has demonstrated that life mulch suppresses weed and is a sustainable strategy to herbicide alternative use, and that the diurnal soil temperature was reduced, which accounts for usefulness of life mulch like watermelon that have dual purposes of yield in agricultural production system, especially in this part of the world, because it generates income for the poor farmers and for sustainability, for poverty alleviation. Next is the methodology that we used. The experiment was conducted, as I told you, in the farm office, in Nyssen, and the coordinate is as presented in the Ibadan headquarters. And it was carried out during the October season, the off-rain season period, or the dry, what we call the dry season. The watermelon density we use is 1.5 by 0.5 meters. Another density is 1.5 by 0.9 meters. Another one is 1.5 by 0.5 meters. Naturally occurring weed population was used, and weeding was done 3 weeks at 3 weeks interval. And our results is here presented. Average over the 2 consecutive cropping seasons, amaranth and watermelon yields were highest at the density of watermelon of 1.5 by 0.9 meters. And these values are within the range reported by David in 1997 in Crop Protection Journal. And the lowest mean light penetration of live mulch was 0.29 and 0.22% in the first and cropping season. This light penetration is the global penetration index, where we measure by light-cold instrument technology the global radiation compared to the radiation that reaches the floor of the ground of the crops, so that we know the amount of radiation reflected or refracted, so that you can compare your results with global technology. All mulch density transmitted less photosynthetic photoflux density, that is the radiation, relative to the control where you don't plant any crop. And that maximum soil temperature was reduced by amaranth and watermelon in intercrop. Next is the result in tabular form, so that we don't take more space. I will have presented some graphics and some videos, but in the interest of time and the data are presented in tabular form. This table 1 shows the yield of watermelon as well as grain amaranth when intercropped, so that you can compare the yield of these intercropping systems in poverty alleviation vis-a-vis climate change adaptation strategy. The check plot is where you don't plant the watermelon, which is what we use as control, compared to only planting the grain amaranth in a monocropping system. And these are the yields that we got in various cropping systems. As I said, optimal was attained at 1.5 meter by 0.9 meters. And this is the yield of that watermelon in that range. And this is the yield of grain amaranth in that year. Our subsequent presentation, we sum up these yields as well as the income we get, so that you can see vividly the adaptation strategy to climate change that we are talking about. Next result presented is the weed density, as well as the weed biomass with watermelon used as live mulch and their grain, amaranth as intercrop. This check is where you use control, and this control compares with the check, and these are the various densities of watermelon. As you can see, the wheat biomass varies. This has the highest amount of wheat biomass, wheat density, because you are not planting watermelon. And this density of watermelon compared to other ones, suppresses yield. This is what this data is telling you. Next is the technology, measurement of radiation, photosynthetic photoflux density. I've tried to explain the technical realities as a footnote. Compared to what other researchers has done around the world. But in essence, the light transmission was reduced by this intercropping system in the tropics. And Otima was pinpointing to 1.5 meter density of watermelon by 1.9 meters of watermelon. And these are the results. You can see the mean when we compare the first and the second cropping season, that this density is pointing towards this mean of planting watermelon at 1.5 meters by 0.9 meters under a cropping system in the tropics. Next, we now see the soil maximum and minimum temperature in adapting to climate change. Also, when you intercrop in mixed cropping system, you find out that this adaptation strategy of cropping system minimizes the soil temperature for climate change adaptation. And this density is very good when you compare the amplitude. The variations of soil temperature is what we mean by amplitude. And next is the partial budgeting. Agronomically, I try to compare of what economic importance, what we are talking here practically. And then we find that the land equivalent ratio, and in other words, how does the land value when you are cropping, and the cost benefit of the cropping system shows that amaranth green plus watermelon fruit yields peak at the density of 1.5 by 0.9 meters planting density, which corresponded to $5, $5.2 cropping production cost, and $68 gross benefit. The highest net revenue was $7 at the density of 1.5 by 0.9 meters watermelon planting staging. When you compare all other cropping systems, we find that net revenue. And I put some footnote how we derived that. Next is the partial budgeting. Partial budgeting speaks of what you put in economically and what you gain economically. And we find out in different irrigation systems, because this was done in the off-season or dry season period, that the drip irrigation system compared to the drainage channel system of irrigation, the sprinkler irrigation system, the watering can irrigation system, bucket, and then basin, the shaft doof type of irrigation system. When we compare this type of irrigation system in the off-season, the drip irrigation system, the trickle irrigation system are found more beneficial in terms of cost-benefit ratio, and I highlighted them in red. How we derived all this data are put in footnotes. We choose the off-season, dry season irrigation system because amaranth horticultural crops, they sell better, they are more profitable in the off-season system in this part of the world. And this is a good example for other tropical regions. Conclusion, for policy implementation, average over the two consecutive cropping systems which we practice here, amaranth green and watermelon fruit yield were highest at the density of 1.5 by 0.9 meters plant spacing. Not that other plant spacings are not beneficial, but the peak of the optima was found at this spacing. And we found out that all mulch density transmitted less photosynthetic follow flush density relative to the control of other crops, or where you don't even crop at all. In other words, the more dense the intercropping system, the better light is cut off from the weed, and we find the optimal density where up that and below which crop yields and benefits are not as beneficial. And this optima was found at 1.5 by 0.9 meters of density. The soil moisture content was significantly greater in the live mulch treatment when you compare it to where you don't crop the bare soil. Our investigation as well demonstrated that live mulch suppresses weed, had greater light extinction, and lower diurnal soil temperature amplitude that accounts for usefulness of live mulch in cropping systems. And this offers opportunities in adaptation of agricultural production system. Thank the Nigerian government, as well as the International Fund for Science in Stockholm, Sweden, that sponsored this research, and my mentor, Dr. Martin Connaughton of US. Yes?
Yes, go ahead.
Dr. Martins, as well as Professor Dr. Isaac Ayelagbe of the University of Agriculture, as well as the board of the International Society of Horticultural Science. I want to extend my gratitude as well and appreciation to Dr. Annalise, the coordinator of this conference, the LOC, and their sponsors, as well as a host of others. Thank you for your inputs. And I want to stop here.
Okay.
Thank you for listening.
Thank you very much, David. Stay with us because there are some questions. And the first question is if there are other horticultural crops that could be intercropped with watermelon.
Yeah, thank you. There are other horticultural crops, soybean, mucuna. We have about, in the sponsored project, we planted about 5 crops, but watermelon was singled out because I'm presenting for horticultural presentation. There are other benefits of other crops which we tried out, and that's meant for other audience. And that's why I put my emails as well as my contacts for further discussion. But we found out that in this particular case, watermelon, it has dual purpose for the dry or off-season period, whereby the farmers can gain benefit of watermelon in the dry season, which is more lucrative, as well as the grain amaranth that has a high protein content in the cropping system. In other cropping systems like that of mucuna and soybean and other nitrogen-fixing crops, there are other benefits, but for attending to climate change adaptation, we choose this particular case to address climate change scenarios.
Thank you. Another question is that you have been looking to different treatments, but did you also look to different, let me say, soil factors that contributed to yielding these differences? What can be the nutrients, or what can be the responsibility of nutrients or soil water and so on, or soil organisms in the high productivity if you make the optimal combination?
Thank you. This research is a stepwise research. We have broken it up into various aspects. The first aspect I'm reporting here is that of the combination of crops, the cropping system strategy to address the climate change. I'm working further on the, being an agronomist, I'm working further on soil amendments, Right now, I'm interested in biochar and other soil amended organic fertilizers, the one we develop here in the tropics in Nighthawk, compared to other commercial fertilizers in improving this cropping system that we have derived. And that will be presented to other audience, and that's why I've given my contacts.
Okay, and then I have also a question for you, and that is you are doing an intercropping with the main aim to increase the amaranth production. But finally, if you would bring your harvest to the market, is it the amaranth that is going to give you more revenue, or is it the watermelon that will give more revenue?
Thank you for that question. It depends on the season. It depends on the demand. In the dry season period, the watermelon is more lucrative. In the situation where the nutritional aspect is the poverty, grain amaranth will give you more profit, more benefit, because grain amaranth is the highest grain compared to even some temperate crops, richer in protein. Grain amaranth is more richer in protein. We use it. We have developed confectionaries for winning baby food, biscuits, and so on. And when you compare the advantage of grain amaranth biscuit winning food for babies and so on, you find that the grain amaranth serves better in this than watermelon and some other grains, even temperate grains and some other tropical grains. So it depends on the situation. If it is dry season, grain amaranth will be more lucrative. I mean, if it is in the off season, dry season, where there is a glut, it depends on the situation.
Okay, thank you very much. Thank you very much, David Ojo from University of Ibadan, Nigeria. So with this, we are winding off this session, this first session of the Vermeylen room. There will be now a coffee break. You will take your coffee from your own coffee pot. You are most welcome to talk to the different presenters during the coffee break period. And there are a lot of breakout rooms. You could go to Dakar room or to Nairobi room and so on, and you can meet the presenters. Okay, let me leave you here, and we will meet later on. Bye-bye.